This comparative transcriptomic study provides a method of developing genetic tools for metabolic engineering in extremophile oleaginous yeasts. Cell factories for biofuels can be a renewable and environmentally friendly alternative to fossil fuels, yet current cell factories are not compatible with processing scale and production ideal settings. In this study, we evaluated 3 oleaginous yeasts, Debaryomyces hansenii, Debaryomyces subglobosus, and Uroea lipolytica. Both Debaryomyces yeasts were chosen as candidates for fuel production because they are very good at consuming depolymerized lignocellulosic biomass, producing fatty acids, and are extremophiles. Uroea lipolytica was chosen because of its current role as a cell factory. All 3 yeasts were subjected to nitrogen starvation, iron deprivation, and 1 molar salt stress, and our transcriptomic analysis was conducted comparing these 3 organisms under these 3 conditions against our control growth condition. From RNA-seq analysis, we found that unlike Uroea, Debaryomyces yeasts do not increase fatty acid production under nitrogen starvation. We also found that Debaryomyces yeasts rewire over 1/3 of their transcriptome in response to cell stress. Weighted gene co-expression network analysis provided a method to group differentially expressed genes in each organism across all conditions. This provided us with modules of similar trending genes, which we were then able to cluster by trends to compare all 3 organisms. We then had 4 clusters of similar trends, and the first cluster had negatively expressed genes in our control, low nitrogen, and low iron conditions, but positively expressed genes in our high salt condition. To confirm these WGCNA results and our clustering method, we performed unsupervised machine learning, specifically t-SNE with a K of 4 to 22, and these trends matched our output from WGCNA, giving us more confidence to analyze these clusters. Using these clusters, we identified the highest expressed genes from the condition of interest in each cluster, and will use this information to mine for genes of interest. We also identified all annotated transcription factors, transporters, and fatty acid genes, and these will be manually sorted through as potential genes of interest as well.